Welcome to this tutorial that describes how the Halo SC-IPS kit from Preferred Cell Systems is used to measure the identity, purity and strength or potency of cord blood, bone marrow or mobilized peripheral blood products used for hematopoietic cellular therapy. After viewing this video, we also recommend viewing the instructional video entitled How to Standardize and Measure Samples in an ATP Bioluminescence Assay. This will allow you to better understand how the complete assay is performed and what the results are telling you. It will then become clear why Halo SC-IPS not only provides you with the most highly specialized data, but is also the most powerful and advanced potency assay available. In this video, we shall now focus on setting up the Halo SC-IPS assay. So what is a strength or potency assay? A strength or potency assay is nothing more than a method to predict the dose of the active ingredients or components that produce an intended response. It's important to distinguish between a clinical response and a clinical outcome. The outcome is usually downstream from the initial clinical response, which, in this case, is the process of cell engraftment. The outcome is therefore the result of this engraftment response. Now, as much as physicians might view the clinical outcome as a potency assay, it cannot measure potency of the product because the response has already occurred. In other words, potency is not performed retrospectively and cannot be used after the fact. This will be discussed again a little bit later. Why is a strength or potency assay so important? Besides predicting the dose and the possible response, a potency assay determines the identity and purity of the cells in the product and whether the manufacturing process is consistent and produces a stable product. It also determines whether the product will fail or be toxic due to improper potency, and therefore whether the product can be safely released for use. These three aspects cannot be underestimated. An assay that does not provide this information is of no use either to the physician or the patient. This is where Halo SC-IPS enters the picture, because Halo SC-IPS was developed to provide all three pieces of information and more. SC-IPS is short for stem cell identity, purity and strength. Halo SC-IPS takes hematopoietic cellular therapy potency testing to a completely new and higher level than has ever been used before. First, it can determine whether the manufacturing process is consistent because it can identify the most important active ingredients in the cellular therapy product. And then it can determine the purity of these components and compare the components for lesser or greater purity. It can measure the potency of the active components and compare them to a preparation of the same cell source and purity. And finally, it can be used to determine if the active components are at a safe and effective dose in order to elicit the required response. And it does all this by providing you with the most non-subjective, rigorous, reliable and trustworthy results ever obtained for hematopoietic cellular therapy products. In order to do this, you as the user must be prepared to consider a completely different methodology to what you've been used to, and the results that will at first appear rather alien to you. But never fear, all will be explained. Now, the term active ingredients or components has already been mentioned several times. What is meant by this term? One of the requirements to measure strength or potency is the need to identify and measure the active components or ingredients. For hematopoietic cellular therapy products, the active ingredients or components are the stem cells, since without the stem cells there would be no response and there would be no stem cell transplantation procedure. Yet it is astounding that not a single processing laboratory worldwide actually tests the functionality of the stem cells responsible for the engraftment response prior to a patient receiving a unit of cells. In any event, the active stem cells constitute less than 0.1% of all the cells present in the bag of cells transplanted into a patient. The stem cell compartment of the lymphohematopoietic system contains stem cells that are hundreds, perhaps thousands of different stages of primitiveness or stemness. All of these stages would be impossible to measure because our technology does not allow it. From a financial point of view, such an endeavor would be prohibitively expensive. Instead, we discovered that measuring a minimum of 2 stem cell populations is sufficient to provide the predictive information necessary. Halo SC-IPS is therefore used to measure 2 of the most important stem cell populations. The first is the primitive lymphohematopoietic stem cell population, responsible for long-term engraftment and reconstitution, and called SC-HPP, or stem cell high proliferative potential. The second is the primitive hematopoietic stem cell population, primarily responsible for short-term engraftment and reconstitution, because it produces cells of all 3 hematopoietic lineages, resulting in erythroid, neutrophil, and platelet engraftment. This is called SCGEMM and is equivalent to the CFC-GEMM population. These 2 populations, which can be accurately and reliably measured using Halo SC-IPS, both exhibit a high degree of stemness and the necessary proliferation potential for engraftment and reconstitution. Since Halo is not a methylcellulose colony-forming unit, or CFU assay, but instead uses suspension expansion culture, or SEC technology, the normal colony-forming unit nomenclature is not used. Nevertheless, these populations only take 5 to 7 days to measure in vitro and provide all the results to make an informed decision. Now, the FDA recommends tests to identify the cells and assess their purity. There are 4 primary tests recommended, and they are all measured as single-point determinations. The first is cell count, usually the total nuclear cell count, or TNC. The second is viability. This value is provided as a percentage and should be above 85%. The question here is, 85% of what? What is the reference value? The third test is the number of CD34 positive cells. This too is often given as a percentage, but also as an absolute number. This is the only stem cell determination made. However, CD34 encompasses many different cell types, not just stem cells. None of the cells are functionally identified, but instead are lumped together as a relative value. And finally, there is the CFU colony count. This may consist of 1 or more different colony types, which are usually also lumped together as a single total colony count, for which there are no reference values. The assay is therefore used as an all-or-nothing assay. Either the cells grow or they don't. And the amount of growth is not important because there is nothing to compare this growth to. Not only are all of these tests performed as a single point determination, but no information is provided regarding the identity, let alone the purity, of the active stem cell components. This is further confounded by the fact that they are all simple and cheap to perform. Most of the information is based on values determined prior to cryopreservation of the cells and not on a thawed cell sample. And finally, physicians are determined to use clinical outcome as their measure of potency. This is a false assumption. As shown previously, clinical outcome is downstream from the intended response and cannot be used as a potency assay because clinical outcome occurs after the fact, and potency is not retrospective, but predictive. It should be obvious that a single point determination provides no information regarding the identity or purity of the active stem cell components, let alone their potency. Now let's consider a multiple point determination. A multiple point determination simply means that a dose response is performed. Potency cannot be measured using a single point determination. To measure the potency of traditional drugs, the drugs are tested over a wide dose range and their response compared to a reference standard of the same material. A typical dose response for a traditional drug is shown here. It is important that the sample dose response is parallel to that of the reference standard because it indicates that the sample has the same purity as the reference standard. If the sample dose response is to the right of the reference standard, the sample has a lower potency. If the dose response is to the left of the reference standard, the sample has a higher potency. The horizontal displacement between the sample and the reference standard dose response curves is the potency ratio, and this is important because it helps determine the dose of the sample. Cells are a little different because the dose response must be linear, and parallelism of the dose response lines has a different implication to that of traditional drugs. Nevertheless, as you will see, like traditional drugs, the potency of cells must be measured using a dose response curve. However, in contrast to traditional drugs, this dose response curve not only allows the strength or potency of the cellular therapeutic to be measured, but also its identity and purity to be determined. Now let's consider first purity. The more purified the population, the better the response and the more sensitive and reliable the signal. So if you're starting with a total nucleated cell or TNC fraction, the response is going to be very low because the cell impurities in the preparation are diluting and masking the response of the active components. Don't forget, this assay uses the most sensitive readout available, namely ATP bioluminescence, which is more than 5,000-fold more sensitive than any CFU colony readout. This means that if the ATP readout is low, it will be almost impossible to detect any statistically relevant results in the CFU assay. If you start with a mononuclear fraction, in which most of the red blood cells, granulocytes and platelets have been removed, the ATP response is going to be much higher and the slope of the cell dose response is going to be steeper. If you start with a purified cell population, such as CD34 positive cells, the response will be even higher and the slope even steeper. In this way, you can immediately ascertain the level of purity of the stem cells in your product. Couple this with the ability to ascertain the primitiveness and therefore the proliferative capacity of the stem cells present in the sample or product. Since the most primitive stem cells also exhibit the greatest proliferative capacity, you would be correct in assuming that these would give the highest ATP response and the steepest slope in the cell dose response. You would also be correct in assuming that this measurement correlates directly with the purity of the cell preparation. Therefore, regardless of the cell preparation, the primitive lymphohematopoietic stem cells would demonstrate a higher response and steeper slope than the slightly more mature primitive hematopoietic stem cells. These, in turn, would demonstrate a greater response and steeper slope than the more mature stem cells and progenitor cells. In other words, the ATP response and slope of the 3-point cell dose response provides you with the ability to quantify both the degree of purity and primitiveness, and therefore the identity of the cells being detected in the assay. But this is not all the assay is telling you. The slope of the cell dose response also quantifies the proliferation potential or capacity of the cells being measured. when the slope of the sample is compared to the slope of the reference standard of the same material and purity, the potency ratio can be calculated. And this is the basis of any cell potency assay, and a stem cell potency assay in particular, which in this case is Halo SC-IPS. Now let's consider the reference standard. All traditional drugs have global reference standards that can be obtained from the US Pharmacopoeia or other vendors. There are no global reference standards for cellular therapeutic products. Nearly all hematopoietic cellular therapy products are now stored in a cryopreserved state prior to use. This also allows cryopreserved reference standards to be established in-house. To do this, Preferred Cell Systems has a separate assay kit called Halo RS. This assay kit is the same as the Halo SC-IPS kit, but includes a vial of cryopreserved cells against which an in-house preparation of cord blood, bone marrow, or mobilized peripheral blood can be compared. The comparison between the reference standard and the in-house prepared sample is tested in exactly the same manner as described in the second part of this instructional video. Normally, the Halo RS assay kit is required only once for each tissue. This is because once a primary reference standard has been established that is similar to or better than that provided in the Halo RS kit, this primary reference standard can be used to establish a secondary and tertiary reference standard preparation. In each case, a batch or lot of cells is prepared, aliquoted into individual vials and cryopreserved, so that every time it is necessary to determine the potency of a new sample, a vial of cryopreserved reference standard cells can be thawed and tested at the same time as the sample. The most recent in-house reference standard is always used to compare to a sample. When aliquots of the most recent reference standard are running low, it can also be used to establish a new reference standard. In this way, the primary and secondary reference standards are available as backups. More about establishing reference standards can be found in the technical manual. Let's begin the second part of this tutorial by taking out the components we need from the Halo SC-IPS assay kit box. Everything you need to culture the two stem cell populations, calibrate and standardize the assay, and measure your samples are in the assay kit box. The assay kit should have arrived frozen, and the box should have been placed in a freezer at -20 degrees Celsius. At this time, you will need to remove the bottle of SC-GEMM Master Mix and the bottle of SC-HPP Master Mix. Also remove the individually wrapped sterile 96 well plate. Everything else can remain in the box at -20 degrees Celsius. You will also need the following supplies: 12 5 ml sterile plastic tubes with lids, single channel pipettes with sterile tips. We recommend using either professionally calibrated manual pipettes or electronic pipettes. The latter are best since they self-calibrate every time they are used. However, all pipettes should be calibrated at regular intervals. You'll need a vortex mixer. You'll also need a humidified chamber, the construction of which can be found in the technical manual. A humidified incubator at 37 degrees Celsius, containing an atmosphere of 5% CO2 and, if possible, 5% O2 is also required. The low oxygen tension reduces oxygen toxicity and increases plating efficiency. Finally, you'll need your sample cell preparation and the reference standard cell preparation. Now let's briefly consider the cell preparation itself. Unless you are using an apheresis mobilized peripheral blood preparation, It is generally routine to use a so-called total nucleated cell or TNC preparation. This is a cell preparation that has usually been partially red blood cell or plasma reduced, leaving a large proportion of cell impurities. Although a TNC preparation can be used in Halo SC-IPS, it is strongly discouraged because the large number of cell impurities mask and dilute the very small proportion of primitive stem cells. Although Halo SC-IPS is about 5,000 times more sensitive than any CFU assay, the low response for both primitive stem cell populations will not provide a reliable and reproducible measure of stem cell quality or potency. These results have been published previously. For this reason, we highly recommend that both the sample and reference standard cell suspensions be fractionated to at least the mononuclear cell or MNC fraction using a standardized density gradient centrifugation protocol. This will ensure that both primitive stem cell populations will be capable of producing an adequate response or signal, providing they are not damaged or dead. Since it is routine to measure cell viability using dye exclusion, make sure this is at least 85%. Having said this, however, even a dye exclusion viability of greater than 95% is no indication that the cells are metabolically viable or will grow. Therefore, for Halo SC-IPS, a dye exclusion viability measurement is actually irrelevant. Halo SC-IPS is based on the ability of cells to produce intracellular ATP in their mitochondria and is therefore a metabolic viability assay. Please also be sure not to base the MNC count on viable cells, as this can result in serious errors. Measure the MNC count directly and perform all cell concentration adjustments on this MNC count. Make sure that your cryopreservation and thawing protocols result in as little damage to the stem cells as possible. This can be monitored using the Halo QC assay kit. Within the tissue sample, there are literally hundreds, perhaps thousands of different cell populations at different stages of development that have the ability to proliferate. These include stem cells, progenitor cells, and precursor cells. The cells with the highest proliferation ability and potential are the stem cells. They are also the ones responsible for engraftment and reconstitution. However, we are only interested in two primitive stem cell populations. All of the other populations are irrelevant. We are also not interested in whether the cells can form colonies in the CFU assay, because if the cells can proliferate, they will differentiate and form colonies by default. A cell suspension of the tissue is prepared and the mononuclear cell count determined. As previously described, the cell concentration is adjusted and a 3-point cell dilution prepared for the sample tissue. For a mononuclear cell or MNC preparation, this is 400,000, 200,000 and 100,000 cells per milliliter. You will only need between 0.3 and 0.4 milliliters of the highest cell concentration. A similar 3-point cell dilution is also prepared for the reference standard, which should be of the same tissue type and purity. We will then have 6 5 milliliter tubes for the sample and 6 5 milliliter tubes for the reference standard. 3 sample tubes will be labeled SCGEMM for each of the 3 cell doses, and the other 3 sample tubes will be labeled SCHPP for each of the 3 doses. The 6 reference standard tubes will be labeled similarly. Once the Halo master mixes for SCGEMM and SCHPP have thawed completely, mix by gentle inversion. Then accurately dispense 0.9 milliliters of the SC-IPS Halo Master Mix into each of the 3 tubes for the sample and the 3 tubes for the reference standard. Follow this by dispensing 0.9 milliliters of the SC-HPP Halo Master Mix into the 3 sample tubes and the 3 reference standard tubes. Now dispense 0.1 milliliters from the lowest sample cell concentration, 100,000 cells per milliliter, into the first SC-IPS and first SC-HPP tubes. This reduces the cell concentration tenfold to 10,000 cells. This is followed by dispensing the second sample cell dose, which is 200,000 cells per milliliter, to the second SC-IPS and second SC-HPP tubes. The cell concentration in this case is reduced to 20,000 cells. Finally, dispense the highest sample cell concentration of 400,000 cells per milliliter to the third SC-IPS and third SC-HPP tubes. The cell concentration will be reduced to 40,000 cells. In each case, you have now prepared the SC-IPS and SC-HPP Halo culture master mixes. Repeat this procedure for the reference standard tubes. Using a single channel pipette, the wells of the first column of the sterile 96 well plate included with the assay kit are each filled with 0.1 milliliters from the lowest SC-IPS sample cell dose. This reduces the lowest cell concentration another tenfold, down to 1,000 cells per well. In other words, you are preparing 8 replicate wells for each cell concentration. Now fill each of the wells in the second column with 0.1 milliliters from the second sample SC-IPS Halo Culture Master Mix. This will reduce the second cell concentration from 20,000 cells to 2,000 cells per well. Fill the third column wells with 0.1 milliliters from the highest SC-IPS Halo Culture Master Mix, thereby reducing the cell concentration from 40,000 cells to 4,000 cells per well. Repeat this procedure for the sample SC-HPP Halo Culture Master Mix. They will occupy the 4th, 5th, and 6th columns of the plate. At this point, half of the plate will now be occupied with cell cultures. Now dispense the reference standard SC-IPS and SC-HPP culture master mixes so that at the end the whole 96 well plate is filled. Replace the lid onto the 96 well plate and place the plate in the humidified chamber described in the technical manual. Transfer the humidified chamber to the humidified 37°C incubator gassed with 5% CO2 and, if possible, 5% O2. Incubate the cells for 7 days. Incubation can, if necessary, be stopped at 5 days. However, between days 5 and day 7, there is a 2 to 3-fold exponential increase in stem cell proliferation. This results in a 2 to 3-fold increase in sensitivity of the assay. Now let's take a look at the results you might get. The form of the results will be similar to that described in the section on multiple point determinations, so we're repeating a little of this. First, plot the 3-point dose-response curves for each of the 2 stem cell populations and their reference standards. Next, fit each of the 3-point dose-response curves to a linear regression. If your instrument software does this for you, great. If not, you can use Excel, GraphPad Prism, or any other statistical software package that gives you the regression parameters. The amount of ATP concentration at a particular cell dose gives you the proliferation ability, or a quantitative determination of stem cell quality. Now we have to focus on the slope of the graphs. From the linear regression parameters, divide the slope of the sample stem cell dose response by the slope of the reference standard stem cell dose response for both stem cell populations. The result is called the potency ratio. Here are some real-life results using cryopreserved cord blood samples. The first thing to know is that the potency of the reference standard is always 1. If the potency ratio is less than 1, the potency of the sample is lower than that of the reference standard, and a greater number of cells will be required to produce the same response as the reference standard. Conversely, if the potency ratio is greater than 1, the potency of the sample is greater than that of the reference standard, and fewer cells will be required to produce the same response as the reference standard. It follows that your sample, and therefore your stem cell product, should exhibit a potency ratio similar to that of the reference standard or better than that of the reference standard. The combined potency ratio of both stem cell populations, namely SC-HPP and SC-GEMM, should be equal to or greater than that of the reference standard. There are many in-between values and conclusions that are possible. However, this type of graph tells you instantly whether a product can or should be released for use in a patient. As you can see, the results, interpretation, and conclusions far exceed those using any present tests. Halo SC-IPS provides everything needed for a rigorous and reliable potency assay of a stem cell therapeutic from umbilical cord blood, bone marrow, or mobilized peripheral blood. Responsible leaders and most of the field have known for years that present testing does not provide the necessary attributes to guarantee a safe and effective stem cell transplantation therapy for patients. It is still based on trial and error. For more information on the science of why, when, and how to measure the potency of a cord blood, bone marrow, or mobilized peripheral blood cellular therapeutic product, visit our website at www.preferredscellsystems.com/haloscips.php. There you will find articles to download, a description of results, what they mean and how they are interpreted, an assay ordering kit information, and much, much more. Preferred Cell Systems is the only company in the world that specializes in application-specific assays for all stages of the hematopoietic stem cell therapy procedure, from the time a potential product reaches the processing laboratory to monitoring patients for reconstitution after stem cell transplantation. These assays include the progenitor cell assays, or PCAs, which are reliable and more applicable than any methylcellulose CFU assay. StemPredict, which is the most rapid stem cell quality assay on the market today. It allows the processing laboratory to triage low from high quality stem cell products in just 3 days. It can be used on fresh or frozen samples. Halo QC, which is used for optimizing procedures such as cell depletion and purification, cryopreservation and thawing, to ensure that the stem cells have not been lost or damaged. The TE, or time to engraftment assays, can reliably monitor short-term engraftment before acceptable numbers are seen in the circulation. And finally, the patient monitoring after transplantation, or PMT assays, which offer a global view of hematopoietic and even lymphopoietic short and long-term engraftment and reconstitution. Finally, we assume that you want to know that the product you are producing is of high quality and is going to do the job it is intended for. However, your patients want and need to know that they are receiving stem cells of the highest quality and potency. It is far cheaper to produce a stem cell product of high quality and potency with the prediction that it should engraft than having to treat a patient whose stem cell transplantation has failed due to inadequate quality and potency. Thank you for joining this tutorial. We hope it has been a valuable resource and look forward to being of service to you in the very near future. Please contact us with any questions you might have. Thank you.